Okay, hello everyone. Welcome to biology session, and today we are going to solve some question in biology paper 2. As you know that in advanced level we have 3 papers, biology 1, biology 2, which are theory, and biology 3, which is practical. So for today we are going to start solving question in biology 2, and we are going to start with the question number 1. So we have question number 1, A. The question states that states the lowest classification taxon at which a cobra and a human are grouped together, and give reason to support your answer. So here we are required to state the lowest classification taxon at which a cobra and a human are grouped together, and of course to give supportive reasons as to why the cobra and the human are placed in that taxon. Okay, so before stating the exact taxon, the lowest taxon of human and the cobra, let us see the classification of cobra and the human to the species level. We normally have 7 taxa. We have 7 ranks of classifying organisms. The highest taxon is a kingdom, followed by the phylum, class, order, family, genus, and the last is species. Okay, so now let us see the classification of cobra. So the kingdom of cobra is kingdom Animalia. The phylum of cobra is Chordata. Class Reptilia. Order Squamata. We have the family Elapidae. The genus Ophiophagus, and the species the Hana. For the part of human, the kingdom to which human belong is kingdom Animalia, phylum Chordata, then you have the class Mammalia, then the order Primates, then you have the family Hominidae, and the genus Homo, and the species to which human being belong is Sapiens. Okay, so according to our question, we are supposed to state the lowest taxon where human and the cobra are placed together. Okay, so let us see now the kingdom. Okay, the kingdom of cobra and the human. Okay, both they have the same kingdom, that is kingdom Animalia. Phylum, of course, again they share the same phylum, that is Chordata, but they differ in the other taxas, the other ranks. So cobra and the human, they share 2 ranks. The first one is a kingdom, both kingdom Animalia, and the second one is phylum, both phylum Chordata. So here we have 2 ranks, but we are required to state the lowest taxon. So normally kingdom the highest, followed by the phylum, followed by the class, but here because these organisms, cobra and the human, so they have 2 ranks in common, that is a kingdom and a phylum. So between the kingdom and the phylum, the lowest is a phylum. So the lowest taxon to which a human and a cobra can be placed is a phylum, whereby both cobra and human share the phylum, that is the phylum Chordata. Okay, so the lowest taxon is a phylum, that is the phylum Chordata. And the other part of the question requires us to state the reason as to what, the reason to justify the answers. So the cobra and the human are placed in the phylum Chordata because both the cobra and the human, during their embryonic stages, they have characteristics which are the characteristics of the phylum Chordata. Okay, so let us see the characteristics of chordata. All chordates have mainly 4 characteristics, particularly during their embryonic stage. So the first one is a notochord, as you can see here, is the first. Then we have the second one here is a dorsal hollow nerve cord. The other one is pharyngeal pouches or pharyngeal slits. And the last one is the post-anal tail. So all chordates have these key features, particularly during their embryonic stages. So because the cobra and the human being during the embryonic stages have these key 4 characteristics, that's why they are placed in the phylum Chordata. Okay, so so the reason is because of the presence of notochord. The notochord later on will be replaced with the vertebral column. The hollow dorsal nerve cord later on will form the central nervous system. The central nervous system consists of the brain and the spinal cord. And of course the pharyngeal slits or visceral cleft later on will form the jaw and the inner ear bones. The jaw and the inner ear bones. And of course the post-anal tail in human being will degenerate. In human being will degenerate, but will develop of course in cobra to make a tail, but in human being will degenerate to form a structure that is called vestigial in the adult, of course, human being. So the adult human being do not have a tail. Okay, so this is the reasons to justify as to why cobra and the human being are placed in the same taxon, that is the phylum Chordata. Okay, so that was the question number 1A. Now let us see the question number 1B. So here we have been given a diagram. The diagram below represents a gut parasite of man. This parasite is found inside the gut of a man. So then we have parts here. We have part A, we have part B, we have C and D. So these parasites here, of course, is tapeworms. Is a tapeworm. Normally the tapeworm, they have in their heads they have rows of structures that are called hooks, and of course they have in their head they have other structures that is called suckers, and they are segmented. Okay, they segmented. Of course they have the neck, the neck is not segmented, and they have segmented, they have so many segments. The segments are called the proglottids, and these proglottids, they have names. We have these proglottids near to the neck and the head. They are called immature, and those which are found somewhere in between here, they are called the mature proglottids, and those found at the tail, at the end, are the oldie proglottids, that is called the gravidae. Okay, normally these gravidae, they break down, and these proglottids, normally they have the eggs. So when they break down, if they will enter into the specific host, intermediate host, so they will develop to form a larva. Okay. So the question from that diagram, so name the genus and the phylum to which it belongs. So the phylum is Platyhelminthes. The name is derived from their appearance. Platy means flat, and helminthes means the worms. So these worms have a flat, dorsoventral flattened worms. They are flat at the back and at their frontal side. So the phylum is this one here, Platyhelminthes. The genus to which these organisms belongs is Taenia. Okay, so this is the responses of the part A of question number 1B. And Roman number 2, give the names of the structures labeled A to D. Okay, let us back to the diagram. Let us see the diagram, back to the diagram. Okay, so we have A here. So the structure that is labeled A is hooks. It's hook structures. Of course, normally the hooks, it's like rows. Okay, they are in row. Here there are 2 rows, okay, of structure that is called the hooks. And B here, the structure B is the suckers, and C here is immature proglottids, and this one here is the oldie proglottids, or called the gravidae. So A stand for hooks, B suckers, C is immature or young proglottids, and D is terminal or the oldie proglottids that are called gravidae. Okay, yeah. Okay, let us see the other part of the question. Question number B, 1B, Roman number 3, requires us to briefly explain how the structures A and the structure B adapt the organisms to its mode of life. Okay, let us see the diagram again. So here we have the structure A, that is the hooks, and the structure B, that is the suckers. So how this structure facilitates these organisms to adapt to their host. Remember that these organisms, of course, are parasites, and these parasites normally they live inside the intestine. Okay, so because they live inside the intestine, and in the intestine there, sometimes there is contraction and the relaxation of muscles that help to push, of course, the food. that contraction and relaxation of muscles that is called peristalsis. So, and these organisms, in order for these organisms, okay, to live inside the intestine, they need to have some structures that can help them to attach to the walls of the intestine. So the structure A and the structure B, that is the hooks and the suckers, they help these organisms to attach in the walls, the mucosal layer of the small intestine, and of course they prevent them from being swept away by the process of peristalsis. Okay, so we are saying structure A, that is the hooks, and the structure B, that is the suckers. These structures are used for attachment of they help the parasite to attach on the intestine wall, the mucosal layer, and of course they resist being swept away by peristalsis. Peristalsis is like a continuous contraction and relaxation of the the intestine muscles, and this of course they help to push the food substance. Okay, so this structure A and B, they used as a means of attachment, a means of helping the parasite to attach to the intestine walls. Okay. So the other question, how does now the organisms harm its host? The host of these organisms is a man. Okay, and normally the adult organisms, the adult one, they are found in the intestine. They are found in the intestine. But the larvae, they can be found in the brain, they can be found in the muscle. Okay? So these organisms, of course, the parasites, when they are large in number, particularly intestine, they can block the lumen. They can block the lumen of the intestine. So these organisms, they can cause the intestine to close. They can close the lumen when they are large in number. So they cause obstruction, and of course obstruction to the intestinal lumen. This complication, of course, can sometimes lead to individuals having this complication to undertake, of course, surgery in order to remove the worms. Okay. So one of the complications is that they can close the intestinal lumen, particularly when they are the adults one. The other complication has to do with the larva. So the larva particular of the form of bladder. So the bladder worm cyst, when found in the brain and the eyes, they can form cyst in the brain and the eyes. So once they form cyst, if they are found in the eyes, they can cause blindness, and if they are found in the brain, they can cause epilepsy and even death. Why? Because, as you know, that brain is a very delicate organ. Okay, so these parasites, they can bring complication to the eyes. They can cause blindness when they are found in the eyes, particularly the larval form. The other complication, these organisms, these parasites, they do not have digestive system. So they depend totally on the digested food from their host. Okay? They do not have digestive system, so they depend totally on their host for food. So they get food from the host. So remember that the host, for example, the human, they need the digested food for their uses. So if this organism they will use the digested food from their host, okay, they may result into poor nutrition to their host. So the host will get fewer nutrients. Why? Because some of the nutrients will be utilized by these parasites. So these parasites, particularly if they are large in number, particularly in the intestine, okay, they can lead to the poor nutrition. Why? Because they use the digested food substances. Okay, so this is also the other complication that can be caused by these parasites. As I told you that the larval form, of course, they can be found in the muscles, different muscles. So once they are in muscles, they can cause muscle pain. So they can form cysts, those larvae, they can form cysts in the muscles, which can lead to the muscles pain. Okay. The other complication, remember that these organisms, they use the suckers and the hooks to attach in the walls of the intestine. So if they attach firmly in the wall of the intestine, they can lead to the bleeding, and therefore leading to the shortage of blood, a condition that is called anemia. So they can cause bleeding from the site of attachment. They attach using their hooks and suckers, and once they attach, they can cause bleeding. Bleeding can lead to the shortage of blood to the given host, leading to a condition that is called anemia. Okay, the other complication is that these organisms, once inside the abdomen, they can cause abdominal pain, therefore interfering with the feeding of the host. So this is all about the complication that can be caused by these parasites, the Taenia parasite. Okay. Now let us see the other question. That is question number 2. A. Discuss secondary growth in a typical dicotyledonous stem using the following guidelines. Discuss secondary growth in a typical dicotyledonous stem using the following guidelines. Okay, so the first one, that is Roman number 1, we are required to explain the meaning of the secondary growth. The meaning of the secondary growth. In plants, we have two types of growth. The first one is called the primal growth. The primal growth normally lead to the increase in height either of the shoot or the roots, and the other types of growth in the plants is called secondary growth. Secondary growth normally, this is a kind of growth which lead to the increase in the diameter or thickness of the plants. It can be the thickness of the stem or the roots. Okay, so the one which occur, and normally primary growth take first, followed by the secondary growth. So secondary growth occur after primary growth as a result of the activity of lateral meristems, and result in the increase in the width, diameter or width. Normally, in the plants we have different tissues, but the only tissue, the only undifferentiated tissue is called the meristems. So this meristematic tissue or meristem tissue is a kind of tissue that retains the ability to divide. And as we know, growth depends on cell division. So when the cell division, particularly mitosis, take place, it tends to increase the number of cells, which later on lead to the, of course, growth of parts. The growth can be either in terms of length or in terms of thickness. Okay? And these meristems, of course, we have 3 types of meristems. We have the apical meristems. The apical meristems normally they are those types of meristems normally that can lead to the growth of the shoot or the roots, increase in the height of the shoot and the roots. But the other type of meristems is the lateral meristems. So this lateral meristems, of course, this is a type of meristems that can lead the plant to increase in diameter, thickness. Okay? So the secondary growth normally, the secondary growth or the increase in thickness normally is as a result of the formation of the secondary phloem and secondary xylem by vesicular cambium and cork cambium. So the lateral meristems, we have 2 kinds of lateral meristems. The first one, we have the vesicular cambium, and then we have the cork cambium. Okay, the vesicular cambium will form the secondary xylem and the secondary phloem, but the cork cambium will form the cork cells. Okay, the outermost layer of the stem. So secondary growth the kind of growth that led to the increase in thickness, and normally these kinds of growth take place after the primary growth. Okay, as you can see here in the diagram, so this is a primary growth. Primary growth normally lead to the formation of the phloem and the xylem. Okay, so here as you can see here, we have outside is a phloem, the inside is the xylem. Okay, and this one here is called the cambium, procambium. So in the secondary growth now, what is taking place? Secondary growth normally, the cells that the kind of meristems or lateral meristems that are responsible for secondary growth are the vesicular cambium, this layer here, as you can see here, the vesicular cambium, and of course the cork cambium. So here inside you have the pith, and then you have the primary xylem. Okay, so this vesicular cambium to the inside, this layer here, to the inside will form the secondary xylem. Inside will form the secondary xylem. Outside will form the secondary phloem. The secondary phloem, of course, will push the primary, will push outward the, of course, the primary phloem. So the vesicular cambium being one of the lateral meristem, the one that is responsible for formation of the secondary xylem as well as to the secondary phloem. But the other lateral meristem is the cork cambium, and of course the cork cambium will form the cork cells, which later on will form the cork. Okay, and the cork together with the secondary phloem the secondary phloem will form the back, the outermost part of the hard. Of course, the back is somehow hard. The outermost part of the plants, particularly the stem or roots. Okay. So that was all about the secondary growth. The other part, we are required now to explain the meristematic tissue, to mention or to explain the meristematic tissues involved in the secondary growth. So as I told you that the lateral meristematic tissue, lateral meristematic tissue that are responsible for the secondary growth, they are of two kinds. Okay, we have the vesicular cambium and we have the cork cambium. The vesicular cambium later on will form the secondary xylem to the inside, and to the outside normally it forms the secondary phloem. So the first meristematic tissue that is responsible for secondary growth is the vesicular cambium, which will form the secondary xylem and the secondary phloem. The other one is the cork cambium. The cork cambium, of course, it is the outermost. It is found to the outermost lateral meristem, and normally it produces the cork cells, and of course the pheroderm. The cork cells will form the corks, and the phloem together with the cork cells form the bark, which normally protect the plants against physical damage and helps to reduce water loss. So the cork cambium, the cork cells, the pheroderm are collectively called periderm. The periderm substitute the epidermis in mature plants, of course. Okay, let us see the. So here, as you can see here, we have the vesicular cambium. This layer here, this is the vesicular cambium. The vesicular cambium to the inside, to the inner side, will form the secondary xylem. To the outside here will form the secondary phloem. So inside, secondary xylem, to the outside, secondary phloem. Okay, and the other meristematic tissue that will form the secondary tissue is the cork cambium. The cork cambium here. Okay, so the cork cambium, of course, will form the cork cells. The cork cells will form the cork, and this cork together with the secondary phloem will form the bark. Okay, will form the bark. As you can see here, this one here is the secondary. This one here is the secondary phloem. So the secondary phloem together with the cork, this one here will form the bark. Okay, and a combination of the cork, okay, the cork together with periderm, of course, the cork and the bark normally form the structure that is called the periderm. Okay, so this is another diagram, as you can see here. We have the xylem. The secondary xylem normally, as you can see here, the vesicular cambium. To the inside will form the secondary xylem, and normally the secondary xylem, they form the woody part. For example, the timber and whatever, that is a secondary xylem. And to the outside, of course, the outside vesicular cambium will form the phloem, secondary phloem that will form the bark. Okay, so here you can see here we have the outermost part of the bark, and then you have the innermost, and the inner part of the bark, of course, they contain structure that is called the pheroderm. Okay, yeah, so This is another diagram that explains more about the vesicular cambium, the product of vesicular cambium. Okay. Again, the vesicular cambium normally have two kinds of cells. We have the short cells, they are called Ray initials, and the other cells, they are called fusiform initials. So vesicular cambium have two kinds of cells, Ray initials, and the fusiform initials. We shall see later on. Okay. So here, of course, the other question, we are supposed to explain the development. Let us development of okay. So the other question, that is question Roman number 3. Roman number 3. Normally, I mean, the question of Roman number 3 is not written here. Sorry. Requires us now to explain the formation of secondary tissue. Requires us to explain the formation of secondary tissue. Okay. So, okay. So the secondary tissue, that is the formation, requires to explain the formation of the secondary tissue. That is how the vesicular cambium is formed, the formation of vesicular cambium. And of course, the vesicular cambium and the outcome or the product that will be formed by the vesicular cambium, that is the secondary xylem and the secondary phloem. Okay. So now let us see how this how the vesicular cambium is formed. Okay. So we are saying initially the vesicular cambium is present in the form of a thin strip between the primary xylem and the primary phloem, and this is called vesicular cambium or intravesicular cambium. We shall see in the diagram. And of course, we have the other part that will form the vesicular cambium is present between the vesicular bundle and is called intervesicular cambium. We shall see the difference between intra, here is intravesicular cambium, and this one here is called the vesicular cambium. Okay. Okay. Here, as you can see here now, let us see the diagram here. So this is here. This is the vesicular cambium. So the vesicular cambium is formed by the vesicular cambium. The vesicular cambium, this is a layer between the phloem, and the xylem. Somewhere in between there is a layer. This layer is called vesicular cambium or intravesicular cambium. So intravesicular cambium, this one here. And of course, between one vesicular bundle consists of a xylem and a phloem. So this is vesicular bundle, 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 vesicular bundle. Okay. So between one vesicular bundle and the other vesicular bundle, okay, there are cells, or there is a kind of cambium that is called intervesicular cambium. So the cambium now, the vesicular cambium will be formed by joining the fascicular cambium or intravascular cambium, this portion here. So when intravascular cambium combine with intervascular cambium, so when you join the this portion here, the vascular cambium, when you join with the intervascular cambium and then join with the vascular cambium, so later on you will get a layer that is called the vesicular cambium. Okay, so when we talk about intravascular cambium, we mean the cambium that is between the phloem here, between the phloem and the xylem, and the intervascular cambium is that cambium that is between the vesicular bundle, between one vesicular bundle and the other. This portion here, this one here, is called the intervascular bundle. Okay, so here is another diagram, as you can see here. Okay, so we have the procambium. Of course, this is procambium. Procambium is the other name of intravascular cambium, and of course we have the intervascular cambium. So these cells here, they will divide and they will join with this one here. So the intervascular cambium will combine with intervascular cambium will combine with the procambium or vascular cambium, and that will form a complete layer of vascular cambium. Okay. Okay. Okay, here. So the development of complete cylinder of cambium, normally it is by joining the intravascular and the intervascular cambium. So the joining this can lead to the formation of a complete ring, that is cambium ring or vesicular cambium. And then the activities of cambium ring, that is intravascular cambium, produces a secondary phloem to the outside. Okay, the intravascular cambium normally produce the secondary phloem to the outside and the secondary xylem to the inside. And of course, the amount of secondary xylem produced is much more than the secondary phloem, and this is because the cambium is more active to the inside than to the outside. So the cambium cells divide radial to increase the growth in circumference and tangentially to form the secondary xylem inwards towards the pith. Okay. So the secondary xylem occupies a major portion of the secondary xylem occupies the major portion of the stem forming hard compact mass, the woody. Okay. The original ray initial produce primary medullary layers which run all the way from the pith to the cortex, unlike the secondary which produce medullary ray produced by later initials which passes through the secondary xylem and the secondary phloem. So primary xylem gets pushed toward the center and they become non-functional, of course, and they remain intact. The primary and the secondary phloem gets crushed due to accumulation of the secondary xylem, and the primary is crushed and pushed outwards. So with the increase of the secondary xylem, some of the peripheral tissues also get stretched. It brings about the rupture of the cortex and the epidermis. So this is replaced by the cork that developed from the corky cambium. Okay. So this is a diagram that shows how the cambium is formed. And the answer diagram here, okay. So the last part of this question here requires us to explain the function of the major tissues formed during secondary growth. So during secondary growth, normally we form 4 tissues. We have the rays, or sometimes they are called the ray initials. Then we form the periderm. Then we form the secondary xylem and the secondary phloem. Okay. So now we are required to state the function of the major tissues. I told you that there are 4 tissues that are formed. We have the rays, we have the periderm, then we have the secondary xylem and the secondary phloem. Okay. So the rays or ray initial, this is a help to transport water and mineral salt from the xylem, and of course transport food substance from the phloem radial across the stem. As we know that the xylem normally transport water and the mineral salt. Okay. And the phloem normally transport the food substance. So the lateral parts, the lateral parts of the stem, they need, of course, they need water and the mineral salt. Also, they need food. So they get water and the mineral salt, and of course the food through this, through the cells that are called rays or ray initials. So ray initials, they distribute, okay, the food from the xylem and the food from the phloem to the lateral parts of the whether the stem or the roots. Also, some of the rays are modified to create spaces, and those spaces can be used as a site of gaseous exchange. So gaseous exchange can occur by diffusion through intracellular spaces. Okay. Rays may also be used for food storage, an important function during period of dormancy, as in winter. So the other tissue is periderm. Periderm, it is the outer protective tissue of secondary plant body, which replaces the ruptured epidermis of expanding primary plant body. Okay. And the last one, okay, the last is secondary xylem. So the secondary xylem, normally it acts as a conduction of water and the mineral salts from the roots to the other parts of the plants, and the secondary phloem translocate or transport food substance, the food manufactured in the leaves, okay, to the other parts of the plants. So we are going to end here. According to a time, we are going to end here, but I have some few like suggestion or ideas. So in order to answer questions, particularly in biology 2, first of all, you need to understand the question clearly. Read the question careful in order to understand the question. And of course, because this kind of question, particularly in the paper 2, they carry a lot of marks. They have the marks. The marks in they have a lot of marks. So because of that, you need to provide detailed explanation. So for example, for the part, let's say, of secondary growth, so you will like explain, provide the meaning, okay, and of course the examples. So try to explain in detail, particular to the questions of this paper too. Okay, by saying so, I'm going to end here, and of course we shall meet in the other session. Thank you.